in the life cycle of the Plasmodium, as you can see over here, it's a complex life cycle, so just take one step at a time, and we have discussed all of this so it would be easier. First of all, the mosquito, when the sweet meaty mosquito, it comes and it injects the sporozoite into the human body. Just remember there over here that the mosquito injects the sporozoite into the human body. Now once these sporozoites are injected, these sporozoites reaches the liver cell. As you can see, they reaches the liver cell and they invade the liver cell. So once the liver cells are infected, as a result, what happens that schizon is formed in the liver cell or in the hepatocyte. Now this schizon, when it ruptures, it releases a large number of merozytes. So this cycle of the before the invasion of the RBC, this cycle which occur into the hepatocyte, this is called as exoerythrocytic cycle. Now these released merozytes, these are released into the blood vessel, where in the blood vessel it attacks the red blood cell. Over here you can see this is the RBC. Now this red blood cell is once invaded, and first of all the early trophocyte stage or the ring form is formed. So this early trophocyte stage then develop into a mature trophocyte stage. You can see over here, this is the mature trophocyte stage. So once the mature trophocyte stage is formed, after that a developing schizon, and finally a schizont is formed. So once the schizont is formed, it ruptures and it releases large number of merozytes. So these merozytes will either attack another red blood cell, or some of these merozytes, they develop into the micro and macro gametocytes. So it forms the gametocyte, as you can see over here. Now these gametocytes, they don't cause infection in the human body. So once another mosquito, when a mosquito again comes, a female anopheles mosquito, then it takes these gametocyte and it sucks these from the human blood. So once these are taken by the mosquito within the gut of the mosquito, what happens that the macrogametocyte and the microgametocyte, they complete their cycle over there. So first of all, after their fusion, zygote is formed, that leads to into the ookinete, and after that oocyst is formed, and this oocyst then ruptures and releases a large number of sporozytes. So these sporozytes then enter into the gut wall, and from there into the salivary gland of the mosquito, and from there once again when this mosquito then again comes and bites another human being, as a result it injects the sporozyte again into the human blood, and as again this cycle repeats. So this is called, this cycle in the liver it is called as exoerythrocytic cycle, whereas in the RBCs it is called as erythrocytic cycle, whereas over here in the mosquito this is called as sporogonic cycle. So the exoerythrocytic cycle and the erythrocytic cycle, these are the asexual cycle, or the sporogonic, or these are the schizogonic cycles, the exoerythrocytic cycle and the erythrocytic cycle. So this is the schizogonic life cycle, whereas here in the mosquito it is the sporogonic cycle. So the mosquito is its definitive host, whereas the human being is its intermediate host. So that's all about the life cycle of the mosquito. Now let's wrap it up. The life cycle of the mosquito, we have studied that the asexual and the sexual cycle, they alternate with one another. The asexual cycle, first of all, when the mosquito bites, as a result sporozyte is formed. Sporozyte then infects the liver cells, schizont is formed. As a result, large number of merozytes are released, they attack the RBCs. In the RBCs, early trophocyte is formed, then the mature trophocyte, then schizont is formed. Those schizont ruptures and releases large number of merozytes. Those merozytes then attack either the other RBCs, or they some of them get converted into the macro and microgametocyte, and these are then taken up by the mosquito, where they form ookinete and the oocyst, and the oocyst of ruptures release a large number of sporozyte, which enter the saliva of the mosquito, and through there when the mosquito bites human, again this cycle is repeated. So that's all about the life cycle of the Plasmodium. Let's move forward. Now the pathogenicity of the Plasmodium. In the pathogenicity, we know that the genus Plasmodium will cause the malaria. So all the four types of the all the four parasites, they cause malaria. Now first of all, we have the Plasmodium vivax that causes benign tertian malaria. Tertian malaria, you know that the fever occur after every day, means at the alternative days the fever occur. Then the Plasmodium malaria, which causes the quartan malaria. So the quartan malaria is the malaria in which the fever occurs after every 2 days. Then the Plasmodium ovale, it causes benign tertian malaria again. And then the Plasmodium falciparum, which causes malignant tertian malaria. So this is Plasmodium falciparum. It causes very severe malaria and it has a high mortality rate. So this is the main pathogenicity of it. Now the clinical manifestation. So first of all, the fever, we have said in the tertian malaria or in the quartan malaria, there occur a fever, and that is followed by a period of relief. So in the tertian malaria, this period of relief is 48 hours, and in case of the quartan malaria, this period is 72 hours. So thus in the tertian malaria, the fever occurs at the alternative days, whereas in case of the quartan malaria, the fever occurs after every 2 days. The second thing is anemia. Due to the lysis of the RBCs, what happens that the anemia develops. The next thing is jaundice. Since a large number of RBCs are broken down, large number of pigment is released, so as a result it leads to jaundice. The next thing is a black water fever. Due to the rupture of the RBCs, large number of RBCs, it is mainly due to the Plasmodium falciparum. Then what happens that hemoglobin is released into the blood, into the urine. Now this urine as a result becomes cola colored, or it brown or blackish color. So that's why this is called as black water fever. The next thing is cerebral malaria. Now once the large number of RBCs are broken down, the broken down product, what happens that they blocks the capillary, and as a result large number of the fluid escape from the blood into the tissues, and also there are perivascular hemorrhages. So in the brain it may lead to the cerebral complication such as coma and coma conversion, etc. So this type is called as cerebral malaria, when the cerebral complications develop. The next thing is the retinal hemorrhage may develop the same phenomena. The next thing is splenomegaly or hepatomegaly. So there is due to the large number of the myelocytes in the blood, or due to this parasitemia, what happens that there is a hyperplasia of the reticuloendothelial system, for the resulting from the increased activity in order to deal with the Plasmodium. So as a result splenomegaly occurs, and hepatomegaly is also seen in it. And the next thing is the flu-like symptoms, means headache and nausea, vomiting. So these are also present in it. Here are some symptoms of the malaria. In the central nervous system, it causes headache, it may cause fever, there is fatigue and pain, and as a result chills, sweating, dry cough, and splenomegaly and nausea, vomiting. So these are all the symptoms of the malaria. So these are all the clinical manifestations that you should remember. It may cause fever, anemia due to the RBCs ruptures, jaundice, black water fever because hemoglobin is entering into the urine, cerebral malaria due to the perivascular hemorrhages, retinal hemorrhages, and splenomegaly and flu-like symptoms. So these are the clinical manifestation. Now let's move forward, and the next thing is diagnosis. That how will you diagnose the different type of the malaria? So first of all is the gold standard is to get thick and thin blood smear. So how you get a thick blood smear and how you get a thin blood smear? First of all, a drop of blood is just placed on the slide, so it forms your thick blood smear. So this thick blood smear is usually it is stained with the Giemsa stain, whereas the thin blood smear it is usually stained with the Leishman stain. So and the thin blood smear is get in such a way that a drop of blood is placed over here, and then another slide is placed in such a way that first of all the blood reaches to its edges, and then this slide is moved or dragged by the another slide. So as a result a thin smear is obtained, and then it is stained with the Leishman stain, and then viewed under the microscope, and the various different morphological form that we have studied, so those are seen, and in this way we see that which form of the Plasmodium is causing the malaria. The next thing is hemoglobin level. So hemoglobin level, as you see that the rupture of the RBCs take place, so this will be low. And the next thing is the total bilirubin level, since it is a breakdown product so of the RBC, so as a result it will be increased due to the increased lysis of the erythrocyte. And the next thing is you can do the urine examination. So the proteinuria and oliguria will be seen in the urine. The next thing is the CSF examination. So in the cerebral malaria, the protein will be raised and the glucose level will be decreased. Moreover, for cerebral malaria, you can see now you can do the CT scan for cerebral hemorrhages and edema. So that was all about the malaria. Thank God it ended, and thank you very much for listening. And do subscribe my channel for the further videos. Thank you again.